Good morning, everybody. Today I'm going to discuss about the topic that is the transgenic plants and beneficial traits of the transgenic plant. So first, try to know what is transgenic plants. A plant that contain foreign DNA, that the DNA of the desired quality or gene of the desired quality from outside the plants means from other plant. And how can you transfer? Through recombinant DNA technology. We multiply the transgenic transgenic plants through plant tissue culture. So, in the heading of transgenic plants, do you know examples of transgenic plant? BT cotton. You know BT cotton, BT brinjal. Then golden iron rice. Then flavor saver variety of tomato. That is the examples of the transgenic plants. So in case of transgenic plants, there is a major category scientists have classified the creation of transgenic plant against the stress tolerance. Stress means adverse situation. Do you know what do you mean by adverse situation? Means high temperature condition, high salinity conditions, then scarcity of water means drought condition. In that adverse situation, plant can survive and produce the huge quantity of the fruit, vegetables, cereals. So scientists have developed a different type of transgenic plants. Stress means there are two category of the meaning of stress. Means stress tolerance means the plant that can grow even in adverse situations. First is biotic stress. So try to know what is biotic stress. First, that is the pest resistance. Do you know what do you mean by pest resistance? The plant even grow if there is attack of the insect, attack of the different insect. Even then it can grow and produce huge quantity of fruit, fibers, cereals, vegetables. Next, disease resistance. Second is disease resistance means against different type of virus, bacteria, fungus. And the virus resistance plant also scientists have developed the plant variety and transfer the gene that is resistant to insect, resistant to disease like bacteria, fungus, against the fungus, against the virus. So these are the different gene develop transfer inside the commercial variety of plant. So first we select the resistant gene, identify the resistant gene from the wild variety of plant, and then we transfer that gene inside the commercial variety of plants. So this is the meaning of transgenic plant against bi biotic stress. Biotic means living organism that can damage the plant leaf part, plant stem part, plant fruit, plant cereals. So if the gene is present, resistant gene is present, then the plant can not affected by this insect, then bacteria, fungus, then virus, then means there are different type of the gene that is resistance against different type of pathogen, different type of pest. Again, next category is abiotic. Abiotic means the non-living components that can affect the growth. What do you mean non-living? That is water. So a scarcity of water, that is the drought resistance. So there is some variety of plant like sorghum, jowar, bajra that contain the drought resistant gene. So if you isolate the drought resistant gene from the sorghum, and then you transfer inside the wheat, then the plant can grow even in a scarcity of water. Next is the salinity. Salinity means the alkaline soil condition, means the pH more than 8. Even then plant can grow. Next is herbicide. What do you mean by herbicide? Yes, आप बताइए, कुछ प्रॉब्लम है तो बताओ. So next is herbicide tolerant. So herbicide tolerant means excess use of herbicide can affect the soil fertility. Okay, so even then it can be affect the, even then it can affect the living organism, useful living organism, the beneficial organisms present in the soil. So these are the development of the transgenic plant. Means you develop the herbicide tolerant gene. You put the herbicide tolerant gene in the chloroplast. Yesterday I have discussed the glyphosate example of the herbicide, that is glyphosate, that is Roundup. The trade name is Roundup. So scientists have isolated the gene that is the resistant or tolerant to this herbicide in the crop. So he has isolated from the wild variety. Means the wild means जो जंगल में होता है, जिसका ज्यादा प्रोडक्शन नहीं होता है, लेकिन उसमें जो जीन है वो रेसिस्टेंट है. So scientists have isolated the herbicide resistant tolerant gene and transferred in the commercial variety, in the soybean, in the cotton, means in the means main crop. Then where he has transferred, where the scientists have transferred inside the chloroplast of the plant's leaf. Okay, so that become tolerant. Now if you use the excess amount of herbicide even in the field condition, there is no effect of the cereals crop, only weed will killed by this herbicide. So this is the classification of the transgenic plant, that is the production of transgenic plant against biotic stress and abiotic stress. So please don't talk together, please. आप लोग अपना बात मत करिए ज्यादा, क्योंकि आप बात करो दूसरे को डिस्टर्ब हो रहा है. प्लीज ट्राई टू रिमेम्बर, लिसन. नेक्स्ट next try to listen. Why transgenic plant is essential? Just see the slide. Means the content of the slide, you see nutritional quality. What do you mean by nutritional quality? Just to improve the protein content of the crops, cereals, pulse, like that. Just to improve the vitamin A content, just like example is golden iron rice. Okay. Next, herbicide resistant, means example I have already discussed the Roundup, that is the herbicide, and Roundup ready the variety of the crop of the soybean. So that is the resistant transgenic plant, that there is no effect of the herbicide, even the weeds are growing along that, then weed will be killed by that herbicide, that is Roundup ready. Next is insecticide resistance. So what do you mean by insecticide resistance? Means have you heard about bt cotton? Bt cotton, that is the contain the bt gene. If you compare the normal cotton and bt cotton, so in bt cotton there is a bt gene inside the plant genome. So scientists have transferred this gene through agrobacterium mediated gene transfer techniques, and they have grown the variety, that is the transgenic plant, at a larger scale. So what happens? क्या हुआ कि if there is an attack of cotton bollworm or pink bollworm, that can damage the leaf portion of the cotton or the ball of the cotton. Ball means the fiber along with the seed, that is the fruit of the cotton. So that if the transgenic plant it is BTG cotton, it is, there is no effect of that insect. Why? I have already discussed in the previous class. What happens? Bt gene encode the Bt crystal protein. So generally inside the plant it is in liquid state. When the insect suck it, when this liquid sap enter in the stomach, it becomes solid crystal, and that rupture the membrane of the intestine and kill the insect like cotton bollworm or pink bollworm. So this is the insecticide resistance, that is the Bt cotton variety. Herbicide tolerant, that is the what that is Roundup Ready variety against the glyphosate, that is Roundup the trade name. And nutritional quality, that is the mm, uh, golden iron rice, a variety of rice that contain huge quantity in rich amount of vitamin A to solve the problem of what, that is the deficiency, vitamin A deficiency cause night blindness. So in Asia, most of the children suffering from the vitamin A deficiency lead to night blindness in their early stage. Why? Because vitamin A source is very dear, means maga hai, and everybody cannot afford the purchasing of the carrots or other vegetables. But if the vitamin A content is more in golden iron as golden iron, that is the rice is the staple food of Asian people, and if they are taking only BT means golden iron as, in that situation, then there is no problem of night blindness deficiency disease, and there is no effect on the vision. So vision, that is controlled by beta carotene, and beta carotene is the immediate precursors of vitamin A that help in the vision during observing any objects. Next is biotic and stress tolerance. You have biotic means that come in the insecticidal resistance, means biotic means bacteria, means virus, means fungus resistance. There is so many variety have developed against the fungus, against the virus, against the bacteria for more production of crops. Next is abiotic resistance tolerance. So what do you mean by abiotic? Means the scarcity of water, salinity. Scarcity of water means drought resistance. So if the plant have DR gene, drought resistant gene, then even it can grow in a scarcity of water, no rainfall condition, and produce huge quantity of cereals, fruits, vegetables. Next is salinity. Means even in the saline soil, in the alkali soil, pH more than 8, plant can grow. Next is that is the enhanced shelf life, that is the delay ripening fruit variety, that is the flavor saver variety, and that is the principle, that is antisense RNA technology. Generally what happens, gene that code the mRNA, and mRNA is the sense strand. So scientists have developed the antisense complementary strand of that sense strand, because you know, first gene encode mRNA, and then that mRNA translate into protein, and that protein act as enzyme, a polygalacturonase enzyme that is responsible for ethylene production. So ethylene is responsible for ripening of the fruits. If you see, take the example of the tomato, in that situation, if you stop the production of ethylene, how can you stop? The enzyme responsible for ethylene production, if you can stop, how can you stop? The gene level, you can stop the formation of protein or enzyme that is responsible for ethylene production. So you can enhance the shelf life of vegetables, shelf life of fruit, to improve the market value. It will beneficial for farmer, it will beneficial for seller, it will be beneficial for purchaser, consumer. So that is the enhanced shelf life, means flavor saver variety. It is based on Antisense RNA technology. Industrial products. So many industrial products are produced from the transgenic plant. That is the useful medicinal product, pharmaceutical, vaccine, edible vaccine. There is a concept of edible vaccine. You can produce the vaccine against cholera, vaccine against different type rabies. Means if you are eating the fruit, if you are eating the vegetable, no need of taking extra vaccine, DNA vaccine or conventional vaccine, because antigen is present in that fruit only, and that trigger for production of antibody if you are consuming that variety of the fruit, that variety of the vegetable. तो ये आपको समझ में आना चाहिए क्या क्या एप्लीकेशन है, मैंने बताया है क्या है ट्रांसजेनिक प्लांट्स का. रेसिस्टेंस टू बायोटिक स्ट्रेस, इंसेक्ट रेसिस्टेंस means you can produce huge quantity of cotton fiber. Virus resistance in case of papaya, you can produce huge quantity of vegetable. Fungal and bacterial resistance means the plant that is not affected by fungus and virus in case of vegetable and fruit crops. Next is resistance to antibiotic. Resistance to antibiotic, herbicide resistance, Roundup Ready variety of the soybean or the other in case other crop like corn and other, that is the cotton. So glyphosate resistance, you know, glyphosate resistant variety, that is abiotic resistance. Next is improvement of crop yield and quality, means you can increase the shelf life of fruits like flavor saver variety of tomato. Improve the nutrition, vitamin A content, that is golden iron content in case of golden iron ice. Improved coloration, you can improve the color of the different vegetables just to attract the consumers. So the next is edible vaccine, means if you are eating the transgenic crop that contain the antigen, and if you are eating in sufficient quantity, then there is no need of vaccine against the particular disease. Essential protein, that is enriching the concentration of the protein in the particular legume crops, or essential fat, means you can enhance the fat content of in case of pulse oil producing crops. So these are the application of the transgenic crop. How it is feasible? Through recombinant DNA technology and plant tissue culture. Next, insecticide resistant plant. I have already discussed in the previous class BT cotton. I have told you the source is Bacillus thuringiensis present in the soil. Okay, so this is gram negative soil. हाँ बोलिए, कुछ पूछ रहे हैं आप लोग. अनुष्का, कितने बच्चे प्रेजेंट है आज? अनुष्का, कितने बच्चे प्रेजेंट है? प्रतिभा, नंदिनी, आज तो मैं पक्का घर पे बात करूंगा. अगर आप लोग नहीं चाह रहे हो तो मैं बात करता हूँ. So yes, so try to know the gene, CRY1 gene that is responsible for. बोलिए, आवाज आ रही है क्लियर? So CRY1 gene, try to learn the CRY1 gene, CRY2 gene, CRY3 gene, CRY4 gene. Okay, now this is specific for particular insect. So try to know the gene, what is the name of that gene. Okay, and how it is acting against the different type of insect. Okay, so next is BT cotton. I have already told you the BT cotton, that is the BT cotton generally GMO crop, genetic modified crop. First it was created by Monsanto company, BT cotton. Okay, so BT toxin are insecticidal to larva of the mother moth and butterfly, beetles and cottons, ball and fly, but are harmless to other form of lives. ये जो है किस किस के इंसेक्ट को ये अटैक करता है? लार्वा स्टेज ऑफ द मॉथ, बटरफ्लाई, beetles, cotton. Next, you know BT. What do you mean by BT? B for Bacillus, T for thuringiensis. So try to know what is the BT gene and what is the BT crystal protein. Generally, it is endotoxin. It is endotoxin and kill the insect after rupturing the cell membrane of the stomach. So try to know. So these are the different insect: Helicoverpa, Armigera, Pictophora, Gossipella, Ericia, Vetella. ये आपको समझ में ये मैं शेयर कर दूंगा आपके क्लास. Just see pink wall worm. So that is responsible for degrading larval or caterpillar stage. You can find that will degrade the ball of the cotton. So just see, this is the how they are very notorious insect, and that damage the whole crops in the field. So try to know how it is acting, mechanism कैसे करता है. bt crystal protein that is coded by btg. So what happens when the insect sucking the liquid juice? When the insect, the caterpillar is sucking the liquid juice, then inside the midgut, you will find the toxin are activated to active form by the gut enzyme. Okay, and now activated toxin bind to the receptor. Now it will disrupt the membrane, because inside the midgut means it is going to rupture the membrane of stomach. So this is the midgut membrane, there the alkaline condition, and it becomes solid crystal. So this is the action of bt toxin. Try to know how it is happening. First, it will enter inside the stomach, then it will become solid crystal form, that is the protein, and it will disrupt the membrane. This is the action. Next, I have already discussed the herbicide resistance. That is the Roundup Ready variety. Roundup Ready variety. How it is acting? The gene response. Please, mic को off करो. The gene responsible for resistance or tolerant, we transfer inside the chloroplast. Please off the mic. And next, uh, when we apply the glyphosate Roundup Ready, it will kill the weeds, and the crop is not affected, if, although it is transgenic crop against the herbicide. So generally, Roundup Ready variety we are growing, and there is no effect of the herbicide. So vaccine production, I have already told you the vaccine production inside the banana. You try to remember the banana, tomato, alfalfa, corn, wheat. We use for production of edible vaccine against cholera, rabies, hepatitis. So these are the different model plants for the production of edible vaccine. कोई ऐसा है जिसने माइक ऑफ नहीं किया? ये देखिए,
सर आपके नाम का ही आ रहा है जिसमें
मेरे
माइक
नाम
ऑफ.
का आ रहा है. Next, I want to discuss what are the examples of edible vaccine. अब सुनाई पड़ रहा है? Are you listening?
Yes, sir.
Means remember what are the model plants that is used for production of edible vaccine. First is that is the transgenic tobacco, transgenic potato, that is against the hepatitis B vaccine, and potato also used for production of hepatitis B vaccine, and banana, tomato, alfalfa, corn, and wheat. So what happens? We transfer the gene that is responsible for coding the antigen protein inside that particular crop. And when we are using the fruit or leaf of that plants or cereals of that plants, then automatically there is a production of antibody against that antigen that will protect our body from particular bacteria, fungus, virus. Next is you see the colorful corns just to attract the consumers. That is the example of transgenic corn. And tearless onions. It is a best example. Tearless onion means even you are cutting the onion, you feel tear in your eye. But there is transgenic variety of the onion. There is no production of sulfur compound that cause tear in your eyes. So tearless onion. Next, flavor saver variety. I have already told you what is the basic principle of flavor saver variety. That is antisense RNA technology. So what is happening in case of antisense RNA technology? क्या होता है ये आपको समझना है. देखिए यहाँ पे. So in normal tomato, what is happening? The gene responsible for ripening means the enzyme, that is the protein, is coded by the gene, and that is it will first make the mRNA. So that is PG means polygalactorinase in protein, that is the enzyme, and that is responsible for ethylene production and help in ripening of the quick ripening of the tomato. So scientists have developed the antisense, the complementary sequence of this mRNA. Try to understand. So gene is present in the tomato, you know, and the gene that code the mRNA contain the gene for polygalactorinase. Polygalactorinase, that is the protein. That protein act as enzyme for ethylene production. So ethylene is responsible for ripening. Now scientists have developed antisense, the complementary sequence of that, and insert in the plant. So what happens when there is a formation of the mRNA level? Then antisense mRNA, it will hybridize. If there is a hybridization, no translation occur. If there is a hybridization, no translation occur because it is busy in hybridization. Synthetic antisense construct inserted in the transgenic tomato so that there is no formation of the polygalactorinase protein that act as enzyme responsible for ripening of the tomato. So that is the concept antisense RNA technology. So first scientists have developed the antisense construct of the gene of the enzyme polygalactorinase enzyme that responsible for ethylene production. So what they have construct? Antisense, complementary. Antisense means complementary of that sense strand of mRNA. If they are going to hybridize, in that situation, they no translation, formation of polypeptide chain, that is known as translation from the sense strand. If this is feasible, then quick ripening of the tomato occur. But if it is not feasible, then no production of the this polygalactorinase enzyme that is responsible for ethylene production. So this is the concept of antisense RNA technology. So it is the example, that is the flavor saver variety of the tomato. So try to know flavor saver variety कब ये किया गया था 1992 में, और ये जो है इंट्रोड्यूस हुआ था 1994 as a variety. Okay, so means PG means polygalactorinase enzyme that is responsible for ethylene production. ये क्या प्रोसीजर है, क्या कैसे बनाते हैं, इसमें लिखा हुआ है आइसोलेशन ऑफ डीएनए फ्रॉम टोमेटो प्लांट एंड दैट इनकोड द enzyme polygalactorinase, and that transfer of this gene to the vector bacteria for production of complementary antisense. Okay, and then that antisense into a fresh tomato to produce the transgenic tomato. मीन्स पहले हम इसका एंटीसेंस कंस्ट्रक्ट बैक्टीरिया में डाल के बनाते हैं थ्रू क्लोनिंग मेथड्स. अब जब बन गया क्लोनिंग एंटीसेंस कंस्ट्रक्ट, देन वी ट्रांसफर इनसाइड द सेम टोमेटो, देन टोमेटो बिकम ट्रांसजेनिक टोमेटो. तो ये आपको समझना है, ये कैसे बनता है ये आपको समझना है. और इसका एडवांटेज क्या है? स्लोअर राइपनिंग रेट ऑफ द टोमेटो और एनी क्रॉप दैट हैव सच टाइप ऑफ एंटी कंस्ट्रक्ट. द राइपनिंग लॉन्गर ऑन द वाइन, आपका जो अंगूर है उसमें भी धीरे से राइपनिंग होगा। फुली डेवलप फ्लेवर्स, इंक्रीज द सेल्फ लाइफ, उसको जो आप चाहते हो कि 1 वीक के लिए यूज करे तो आप मैक्सिमम 2 वीक 3 वीक तक ले जा सकते हैं, लेकिन अगर नहीं रहेगा तो 1 वीक में ही ये बर्बाद हो जाएगा, डिग्रेड हो जाएगा। तो दिस इज द नाउ again there is another topic, that is Barnase-barstar system. What is Barnase-barstar system? This is the gene that is the responsible for the enzyme ribonuclease. And what is the function? It will kill the male gametes, pollen grain, and make the male sterile plant, that is female plant. What is the use of the female plant? It will use for hybrid seed production, hybrid variety production. So Barnase-barstar system, it is given in your CBSE test too. That's why I'm dealing with this. This is the what is the source? That is the Bacillus amyloleucophysins. Barnase का जो source है Bacillus bacteria है, and it is cytoplasmic Barnase inhibitor which inhibit the and protect itself, and RNA is linked with the Barnase gene. Okay, so ये आपको समझना है। ये किस में होता है जनरली? ये मस्टर्ड में होता है, कॉर्न में होता है। इट इज ट्राई टू नो व्हाट इज द सोर्स। ये जो है TA29 प्रोमोटर होता है इसमें, जो कि उसके फंक्शन को प्रोमोट करता है, और किसको इनहिबिट करता है? टेपेटल लेयर। अगर आप देखेंगे तो आपको पता चलेगा। देखिए यहाँ पे ये डायग्राम है, ये डायग्राम है जो कि इसका प्रिंसिपल है। सोर्स क्या है बार्नेज का? बेसिलस एमाइलोलिक्विफेसेंस बैक्टीरिया है। ये किसके साथ एक्सप्रेस होगा, फंक्शनल करेगा? TA29 प्रोमोटर। ओके, एंड व्हेन द TA29 promoter is attached with this gene, only then it is functional and it will produce the RNA gene, and it will kill the pollen mother cell, means tapetal layer में degrade करेगा pollen grain को, और फ्लावर में मेल फीमेल पार्ट दोनों है तो किसको डैमेज करेगा? मेल को, so that female will egg will remain viable and plant become female plants. So that production of female plant line, that is used for production of hybrid seed. Now you put the pollen grain of desired characters from the other plant, and then through process of pollination and fertilization we are going to produce the hybrid seed. तो ये आपको समझ में आना चाहिए कैसे ही अटैक करता है बार्नेज बारस्टार जीन। सो बार्नेज जीन इज मेक द स्टेराइल ऑफ द पोलन ग्रेन, एंड बारस्टार जीन दो वर्ड है, एक बार्नेज जीन है, एक बारस्टार जीन। सो बार्नेज जीन इज रिस्पांसिबल फॉर स्टेरिलिटी ऑफ पोलन ग्रेन मेल न्यूक्लियस, एंड बारस्टार जीन रिस्टोरिंग द फर्टिलिटी ऑफ पोलन ग्रेन। तो ये दोनों सिस्टम है। बार्नेज जीन to make the female plant line for making the hybrid variety of particular crops, so that we can get improved character of the particular variety of the crops. तो पहला क्या है कि आप फीमेल प्लांट बनाना है, और बाहर से पोलन ग्रेन जो डिजायर क्वालिटी का उसको लेके उस पे डालेंगे, एंड देन वी एक्सपेक्ट दैट दिस इज द हाइब्रिड सीड, and that hybrid seed have huge quantity of protein, huge quantity of oil, huge quantity of carbohydrate as per our requirements of the human consumptions. So just scientists use this system to make a female line. क्या कैसे करता है? पोलन ग्रेन जो बनाता है पोलन मदर सेल, उसमें से पोलन ग्रेन को ये डिग्रेड कर देता है। कौन सा एंजाइम? दैट इज आरएनएस। ये किसके द्वारा कोड होता है? बार्नेज जीन। और ये किसके हेल्प से फंक्शनल आता है? TA29। So what is the source of Barnase gene? That is the Bacillus amyloliquefaciens, that is the bacteria. और TA29 promoter is the source is from tobacco. तो ये टोबैको में TA29 होता है, और ये बार्नेज जीन होता है कहाँ पे? Bacillus amyloliquefaciens। So it will attack the RNase, it will attack where? The pollen grain मदर सेल, जहां पे पोलन ग्रेन प्रोडक्शन होता है। तब ये बाइसेक्सुअल फ्लावर है, उसमें से पोलन ग्रेन में से पोलन फर्टाइल नहीं रह पाएगा, स्टेराइल रह जाएगा। ओके, सो बारस्टार जीन, बार्नेज जीन क्या करता है? स्टेराइल बनाता है, और बारस्टार जीन जो है फर्टाइल। जस्ट सी ये डिफरेंस इन दिस दोनों स्लाइड में क्या डिफरेंस है? बार्नेज जीन जो है ना स्टेराइल बनाता है, और बारस्टार जीन जो है फर्टाइल बनाता है। सो ट्राई टू नो व्हाट इज हैपनिंग इन केस ऑफ बार्नेज बारस्टार सिस्टम। आपको सोर्स जीन का याद रखना है। बार्नेज जीन स्टेराइल बनाता है और बारस्टार जीन फर्टाइल बनाता है और दैट इज रिस्पॉन्सिबल फॉर हाइब्रिड सीड प्रोडक्शन। ये हाइब्रिड सीड बनाने में ये करता है। और ये कैसे काम करता है ये आपको समझना है कि ये TA29 के प्रोमोटर के प्रेजेंस में ही ये एक्शन में आएगा, अदरवाइज इट विल नॉट कम इन द एक्शन। Means it will disturb the pollen grain male nuclei only in presence of TA29, क्योंकि TA29 help in expression of Barnase gene, और जब तब तक नहीं काम करे जब तक ये TA29 उसमें नहीं रहेगा, and it will destroy the RNA in the tapetum, pollen grain is not produced। और बार स्टार जीन क्या करता है, बार्नेज जीन के एक्शन को रोक देता है। बार स्टार जीन, आई एम टॉकिंग अबाउट बार्नेज जीन एंड बार स्टार जीन। सो बार स्टार जीन इनहिबिट द एक्शन ऑफ द बार्नेज जीन एंड मेंटेन द फर्टिलिटी। ये जस्ट अपोजिट वर्क है। तो आपको समझना है कि बार्नेज जीन बैसिलस एमाइलोलिक्विफेशन बैक्टीरियल सोर्स है। ट्रांसजेनिक प्लांट किसमें होता है, टोबैको है, जो ब्रासिका नेपस मस्टर्ड वैरायटी में यूज होता है। और TA29 का सोर्स क्या है, टोबैको। So that is used for hybrid seed production। ये आपको समझना है। ये क्या है, गोल्डन आयरन राइस। ये समझ लो कि ये क्या है, गोल्डन आयरन राइस। ये नॉर्मल राइस है, इसमें बीटा कैरोटीन है, जबकि विटामिन ए कंटेंट ज्यादा है और इसमें आयरन कंटेंट भी ज्यादा है। तो इसका why it is important, because it is rich source of vitamin A as well as iron। तो ये ऐसे नॉर्मल राइस में ये नहीं बन सकता है बीटा कैरोटीन, बट गोल्डन आयरन राइस there is a formation of beta carotene। so try to know how it is formed। ये कहाँ है, प्लांट से लिया गया, डैफोडिल्स फ्लावर है उसमें से लिया गया है। दिस इज द पाथवे फॉर बीटा कैरोटीन। ये पाथवे क्या है, बीटा कैरोटीन का है, जो कि इमीडिएट प्रिकर्सर है विटामिन ए का। so that is deficient in that is this gene, daffodils gene, then bacterial source gene, that is the phytins desaturate gene, zeta carotene gene, the daffodils gene, that is the absent in the rice। So scientists have transferred the gene through agrobacterium mediated gene transfer technique inside the rice, and then it produce the beta। ये pathway present है, लेकिन ये this step is absent, this step is absent, this is present, दिस इज प्रेजेंट, यहां तक रह जाता है। लेकिन जैसे जीन डाला गया है, ये फ्लावरी प्लांट है, वहां से फाइटिन सिंथेसिस जीन डाला गया है, फाइटिन डिसैचुरेट जीन डाला गया है, बैक्टीरिया से डैफोडिल से फिर से डाला गया लाइकोविन बीटा साइक्लेज, देन देयर इज फॉर्मेशन ऑफ बीटा कैरोटीन। इंगो पोर्टिगस ये सबसे पहले बनाया था, आपके बुक में लिखा हुआ है इसलिए मैं बता रहा हूँ। सो जनरली दैट इज द डिफरेंस इन गोल्डन आयरन राइस एंड नॉर्मल राइस। क्या डिफरेंस है, इसमें रिच अमाउंट ऑफ बीटा कैरोटीन है, विटामिन ए है और आयरन है, जो कि किस डिजीज को क्योर करता है, नाइट ब्लाइंडनेस डिजीज को, देन एनीमिया डिजीज को। सो दिस इज द वेरी यूजफुल ट्रांसजेनिक क्रॉप just to overcome the problem of night blindness and iron anemia deficiency in the poor man people in the Southeast Asian country। So this is the it was developed by Ingo Portiga scientist and try to know what is the pathway, what is the gene। 3 जीन है, आपको याद रखना है। 2 जीन बैक्टीरिया प्लांट से लिया गया है और तीसरा जीन जो है बैक्टीरिया से लिया गया है। कौन सा, इर्विनिया कैरेटोफोरा, इर्विनिया कैरेटोफोरा से लिया गया बैक्टीरिया है। तो आपको ये ध्यान रखना है और आपको ये जितना मैं होमवर्क दे रहा हूँ वो कंप्लीट करना है। प्लीज फॉलो द कंटेंट ऑफ द CBSE। क्या क्या है, आपसे क्या क्या पूछा जाएगा, हर बार ऐसा बीमारी नहीं आना है, इसलिए ट्राई टू गो थ्रू द कंटेंट ऑफ द चैप्टर and try to optimize your knowledge so that you will be ready for CBSE exam। okay, so next you can see the human protein you can also produce inside the plant, and next virus resistant protein you can also produce inside the plant। ये फिर से मैं बता रहा हूँ। so these are the different pathway through which we can improve the quality and we can create a transgenic crops in the plants। तो ये आपको समझ में आना चाहिए कि व्हाट इज ट्रांसजेनिक क्रॉप, व्हाट इज हर्बिसाइड टॉलरेंट क्रॉप, व्हाट इज बायोटिक हर्बिसाइड टॉलरेंट क्रॉप, व्हाट इज ए बायोटिक हर्बिसाइड टॉलरेंट क्रॉप, व्हाट इज द बीटी कॉटन्स, what is the golden iron rice। try to know what is the gene, what is the function, what is the application of, what is the uses of that transgenic crops। तो आपको समझना है और होमवर्क आप टाइम से कंप्लीट करेंगे, अदरवाइज यू विल फेस प्रॉब्लम इन कमिंग एग्जाम्स। तो मैं एक क्विज लूंगा इस चैप्टर का, और आपको समझना है, आप यू गो थ्रू दिस चैप्टर। मैं आउट ऑफ चैप्टर कभी नहीं जाऊंगा। So you follow the chapter line by line and try to know who is the father of golden RNA, what is BT gene, what is the source of BT gene, okay, and what is the advantage of using BT cotton? It is eco friendly, ये कैसे eco friendly है ये आपको समझना है, okay. So now yeah, you will be ready for to next day I will take a test, okay. So try to read the chapter and try to ask the question if you are facing any problems. कुछ पार्ट अभी बाकी है, ऑर्गेनोजेनेसिस, सोमेटिक एम्ब्रियोजेनेसिस। मैं 2 स्लाइड शेयर करूंगा, 2 पीपीटी, उसको आपको गो थ्रू दैट्स पीपीटी एंड ट्राई टू नो। अगर कुछ समझ में नहीं आता है तो आप हमसे पूछ सकते हैं, ओके। तो कितने बच्चे हैं जल्दी से प्रेजेंट आप दे दीजिएगा, क्लियर? हाँ, ओके।
ओके।
सर,
हाँ बोलो बेटा।
अटेंडेंस ग्रुप में देनी है?
हाँ अटेंडेंस ग्रुप में दे दिया मैंने, स्क्रीनशॉट ले लिया है। जिसका जो प्रेजेंट नहीं रहेगा उसको मैं अटेंडेंस नहीं दूंगा। जो मान लीजिए ये ग्रुप में अभी ऑनलाइन क्लास में नहीं रहेगा तो उसको मैं अटेंडेंस नहीं दूंगा। मैं ऑब्जर्व करता हूँ कि कब कौन कौन आता है, कौन 1 मिनट के लिए आया, और ये ऑब्जर्व ये सॉफ्टवेयर भी करता है, 1-1 मिनट का हिसाब रखता है। जो 1 मिनट के लिए है उसको भी रिकॉर्ड करेगा, जो 1 सेकंड के लिए आएगा उसको भी रिकॉर्ड करेगा। सो डोंट ट्राई टू एस्केप फ्रॉम द क्लास। ये बायोटेक भी 100 मार्क्स का और केमिस्ट्री भी 100 मार्क्स का है, और तुम्हारा बायोलॉजी भी 100 मार्क्स का है। सो यू हैव टू डिसाइड योरसेल्फ। आपको डिसाइड करना है कि किस में ज्यादा मार्क्स लाना। 95 जो पढ़ा है उसको 95 आया है, जो नहीं पढ़ा है उसको 70 आया है। तो दिस इज द डिफरेंस। अगर चाहते हैं आप सभी को हमको 95 आए, 96 आए, तो आपको आएगा। सो ट्राई टू रीड द चैप्टर, ओके।